Thank you very much, David. I hope you can all hear me here from Copenhagen. Good afternoon. First, I would like to thank you, David, for inviting me to present at your annual Muscle Health Awareness Day. And I hope and wish that you will have a great day. So as you already mentioned, I will present some data. It will be partly published data and also some unpublished data showing that IL-6 is a physiological regulator of adipose tissue mass in humans. But before I get to that, I would like to say some words about my workplace, which is the Center for Physical Activity Research at the University Hospital in Copenhagen. The center is headed by Professor Bente Klarlund Petersen, and consists of 9 research groups, which all work on different aspects of exercise and its influence on health and disease. And the groups have quite different ways of working, which is supposed to be illustrated in these 2 schematics. So we span somehow from molecular work, basic science, over human integrative physiology studies, also to clinical intervention studies. And then we also engage in more field activities, so participating in the municipalities and hospitals, and also try to change public health related praxis by influencing education and being present in the public debates. So it's a broad thing that we're doing. So my own group, we're a small group, we perform integrative physiology studies, and sometimes we also have a more clinical approach. So we belong to these the blue circles and the blue areas on these schematics. And I'll be frank and say that I think we are quite narrow-minded in my group, as we primarily focus on IL-6, not to say only on IL-6. And we're very much focused on trying to understand how IL-6 is involved in regulating energy metabolism. So I will start by this famous data and say that it all started more or less 20 years ago when this paper was published in Nature Medicine, and it showed that whole body IL-6 knockout mice, they developed obesity with age, and along with that also insulin resistance and glucose intolerance. And around the same time, scientists in Copenhagen, they showed that a single bout of exercise results in increased levels of IL-6. So here on the left, you see an example from a marathon run where IL-6 levels went from below 1 picograms per mil before the marathon run to around 80 picograms per mil post-exercise. So, but as I will show you a bit later, such a dramatic increase is not really what we observe, or it's not necessarily what we observe during a more regular exercise bout, where IL-6 levels typically increase about 5-fold. But the fact that exercise was associated with increased systemic levels of cytokines, that has previously been shown. So the real contribution from the Copenhagen team was that skeletal muscle was the source of exercise-induced IL-6. So with that finding, a whole new paradigm developed, and skeletal muscle was now suddenly considered an endocrine organ, producing and secreting myokines. And these could act probably in an autocrine, paracrine, and also in an endocrine manner to regulate the function of other organs. So during the years that followed, many studies were performed in both mice and humans, trying to understand the physiological role of this exercise-induced IL-6. But at this time, there was also an interest in understanding the role of IL-6 in low-grade inflammation, as was seen in obesity and in diabetes. In Mark Donner's laboratory in Zurich, where I did my PhD back then, we worked on islet inflammation in type 2 diabetes. And we therefore also had an interest in understanding how IL-6 in that context influenced the islet function and glucose homeostasis. So in a number of cell and mouse studies, we found that IL-6 stimulated GLP-1 synthesis in the intestinal L cells, but also in the pancreatic islets, and that this led to an increased glucose-stimulated insulin secretion and glucose tolerance, improved glucose tolerance. So in mice, at least, we showed such a crosstalk between organs. And in this case, it was specifically between skeletal muscle and intestinal L cells and pancreatic islets. And when we tried to take this finding into a human setting, we did so by infusing recombinant human IL-6 into humans, and found, similar to in mice at least, that IL-6 improved glucose tolerance. However, this improved glucose tolerance was mediated by a delay in the gastric emptying rate rather than an increased insulin secretion, as we had observed in the mice. So until 2010 or so, the only way to study IL-6 was to infuse it, at least in humans. The only way to study it in humans was to infuse it. And only a few groups in the world performed such experiments, and most of them were performed actually in Copenhagen. And in the early studies, it was shown that infused IL-6 regulated lipid metabolism by stimulating the mobilization of fat, as well as the use of fat as energy source. But somehow the limitation of these studies was that IL-6 was infused, and that plasma levels of IL-6 were often higher than what one could expect in response to an exercise bout or in a physiological setting. So therefore, it was somehow good news when the IL-6 receptor antibody tocilizumab back in 2010 was approved to treat patients with rheumatoid arthritis. So this gave us the option to obtain a loss of IL-6 function model in humans, one could say, and to study the more physiological role of IL-6. The antibody, this IL-6 receptor antibody, it binds both the membrane-bound and the soluble form of the IL-6 receptor, so therefore it entirely blocks all endogenous IL-6 signaling. And given the consistent effects of infused IL-6 on fat metabolism, one of the first questions we asked was the following: does endogenous IL-6 have a physiological role in regulating exercise-induced changes in adipose tissue mass in humans? And we addressed this question by performing a randomized controlled trial in 56 abdominally obese men and women. And the participants, they were first randomized to receive either placebo or IL-6 receptor blockade. And for the following 12 weeks, half of the participants in each of these two groups performed supervised endurance training 3 times weekly, and the other half served as non-training controls. And before and after the intervention, we performed MRI and DEXA scans to assess visceral and cardiac adipose tissue mass. That was done by MRI, and then lean body mass by DEXA. And we also assessed cardiorespiratory fitness and performed all glucose tolerance tests and mixed meal tolerance tests, and then we also took some tissue biopsies. The primary outcome of this study was change in visceral adipose tissue mass, and this is what you see in this figure. On the y-axis, you see the delta change in visceral adipose tissue mass, and on the x-axis, we have the 4 groups. First, the no exercise placebo group, and then the no exercise tocilizumab group. And both of these groups, they showed a numerical increase in visceral adipose tissue mass over the 12-week intervention, but there was no difference in change between the two groups. And I should maybe mention that if we look at within group changes, there was a significant increase within the group that received tocilizumab. So visceral adipose tissue mass expanded in this group when compared to its pre-intervention levels. As expected, visceral adipose tissue was reduced in the exercise placebo group. And most interestingly, this effect was completely abolished in the group which combined exercise with IL-6 receptor blockade. So exercise-induced reductions in visceral adipose tissue mass were completely dependent on IL-6 signaling. Or said in other words, if an abdominally obese inactive person starts to perform regular exercise, the reduction in visceral adipose tissue mass is completely dependent on the presence of an IL-6 signal. We also assessed changes in cardiac adipose tissue, and we're most interested in changes in epicardial adipose tissue, as this fat depot has been associated with increased cardiovascular risk. But changes in the epicardial adipose tissue were very similar to the changes observed in visceral adipose tissue. So while there were no changes in epicardial adipose tissue mass in the two no exercise control groups, the first two groups here, the exercise placebo group showed a significant reduction, a 30% reduction in epicardial adipose tissue. And this effect was again completely abolished or prevented by IL-6 receptor blockade. So again, to conclude with an image, when an abdominally obese person goes from being inactive to active, a reduction in epicardial adipose tissue is to be expected. But similar to visceral adipose tissue, this effect was dependent on IL-6 signaling. So I would say this left us with the obvious question, how does IL-6 regulate adipose tissue mass in humans? What is the underlying reason for this? And currently we are approaching this question with the hypothesis that endogenous IL-6 regulates substrate turnover in some way, which then potentially can lead to expansion of adipose tissue mass in humans. And we recently finished our first study testing this hypothesis. It was a study focusing on what happens to substrate turnover during rest and exercise, and also during the post-exercise recovery phase, if we block endogenous IL-6 signaling. So the data I'll show you now and onwards are all unpublished. In this study, we included lean and obese men. They were young, between 20 and 30 years of age, and they had a BMI, the lean group averaged 23, and in the obese group, the average was 34. They came in for 3 nearly identical study visits. On all study days, we infused stable isotope tracers throughout the study day. And the stable isotopes were labeled substrates, which included palmitate, glycerol, glucose, phenylalanine, and tyrosine. And on the first study day, we infused saline as placebo, so this serves as our control day. We kept the participants in bed for a 2-hour run-in phase, so that the stable or the isotopes, they could reach a steady state of equilibrium. And then there was a 90-minute resting phase, followed by a 90-minute exercise bout on a bike, and then a 60-minute recovery in bed. And throughout the study day, we collected blood and breath samples, and these were used to determine substrate turnover or kinetics, and also concentrations of substrates and hormones, and also oxidation of substrates. We also performed some indirect calorimetry measurements to assess whole body substrate oxidation in all phases. On the second day, which we call day 1, participants were infused with the IL-6 receptor antibody tocilizumab at the beginning of the study day. So on this day, we are evaluating the acute effects of blocking IL-6. And then on the third study day, which took place 21 days after the infusion of the IL-6 receptor antibody, the participants were still under the influence of the antibody, and we therefore here evaluate the more long-term effects of IL-6 blockade. So to the data, and since all or most of the data slides look like this one, I'll take you slowly through this first one. So what you see here are the concentrations of IL-6 in the circulation in the lean group on the left side and in the obese group to the right. And first, maybe I can mention that there was no difference in IL-6 levels in the lean and obese participants, which is probably due to their young age, so a short life living with obesity. The black line and the dots here represent IL-6 levels on the saline day. So you can see that exercise induces a small increase in circulating IL-6, and I know this may look rather modest, but given the mode, which was spiking of exercise, and also the intensity, which was low intensity, this small 5-fold increase in IL-6 is what we typically find. The red line and dots represent day 1. This is the day where the participants received the IL-6 receptor antibody. And as expected, blocking the IL-6 receptors led to increased circulating levels of IL-6. And this is thought to be due to an impaired IL-6 receptor mediated clearance. So due to this reduced clearance, you can also better appreciate the exercise-induced increase in IL-6 on this day. On the third study day, shown in green, IL-6 levels were elevated already at baseline. And this is due to the long elimination half-life of the antibody and shows that the antibody is still effective at blocking IL-6 signaling. So much for the setup. Now to the substrates. What you see here are the palmitate concentration data. And palmitate increases as one would expect in response to exercise and exercise bout, and it drops again quickly in the recovery phase. And there's no difference in exercise-induced palmitate comparing lean and obese. But when looking at the effect of IL-6 receptor blockade, we also only see a small tendency towards a lower palmitate levels in the lean group here on the left side. And it only occurs seems to occur in the early phase of exercise and only on day 1, which is the day where we assess the acute effects of IL-6. So not much of an effect of IL-6 receptor blockade on the concentration of palmitate. Here we look at the palmitate kinetics. This is the rate of appearance of palmitate, which is a measure of the rate by which palmitate is liberated from its stores. And exercise increases the rate of appearance of palmitate similarly in both groups. But the interesting part here is that when IL-6 signaling is blocked, both acutely and chronically, the rate of appearance of palmitate is reduced. And it's quite a significant reduction, around 30%. And it's seen both during rest and during exercise, and also during the post-exercise recovery phase. So this means that in the absence of the IL-6 signal, more fatty acids, they stay within the cell. And the way that we see this or view this currently is that that there is more reesterification, intracellular reesterification taking place. But when we compare the effect of IL-6 receptor blockade in the lean and obese group, it showed no significant difference. But I think it's fair to say that it appears as if the effect of the IL-6 receptor antibody is a bit less pronounced in the obese group compared to the lean group. So the mobilization of these free fatty acids into the circulation, they occur as a result of lipolysis. And here we look at the rate of lipolysis, which is given by the rate of appearance of glycerol. And we see that it increases in both groups during the exercise bout. And while there is no effect of IL-6 receptor blockade on lipolysis in the lean group, IL-6 receptor blockade has a small reducing effect on lipolysis in the obese group. I mean, it is small, and I would say the p-values are on the border of significance throughout all the phases, and only become really significant in the early recovery phase. And this is probably due to our low participant number in the obese group. We only managed to include 9 participants, and it could very well be that with more people, this would have seen a little bit more clear. But nevertheless, given that all of the glycerol appears in the circulation following lipolysis, there is some information in expressing the rate of appearance of palmitate and the rate of appearance of glycerol as a ratio. And this is what has been done here. IL-6 blockade lowered the ratio in the lean group, showing that less palmitate gets out of the cell following lipolysis. And interestingly, this occurs both during rest and during exercise. So just to emphasize that this is not a phenomenon only relating to exercise. The ratio, on the other hand, is unaffected by IL-6 receptor blockade in the obese group. And this is because IL-6 receptor blockade both, slightly reduce both the lipolysis, but also the release of palmitate. So palmitate taken up from the circulation is either oxidized or re-esterified. And here you see the palmitate oxidation rate derived from isotope enrichment in the expired breath. And the oxidation rates, they increase with exercise without any significant difference between the lean and obese group. And overall, there was no effect of IL-6 receptor blockade on palmitate oxidation. And also likewise, whole body fatty acid oxidation determined by indirect calorimetry was unchanged with IL-6 receptor blockade. So to conclude, endogenous IL-6 regulates adipose tissue mass in humans. We have shown this by blocking IL-6 signaling using the IL-6 receptor antibody, tocilizumab, and we found that blocking IL-6 signaling prevents exercise from reducing visceral and epicardial adipose tissue mass. And there were signs of tocilizumab on its own leading to expansion of visceral adipose tissue mass. And currently we don't know, but one of the, I would say, the many questions that we are left with is whether IL-6 regulates the overall expansion of adipose tissue mass. And we haven't studied this yet. What may be happening is that IL-6 specifically regulates against ectopic fat deposition, which is what epicardial and visceral adipose tissue fat mass is considered to be. But this is something we'll have to look further into. We can also conclude that endogenous IL-6 regulates the turnover of fat. And our data show that blocking IL-6 signaling reduces the release of free fatty acids into the circulation following lipolysis. And we really don't know the mechanism underlying this effect yet. And we, as I mentioned during the talk, we speculate that this is due to an increased intracellular re-esterification. But we will have to do more cell work to clarify that. And then we would also need to understand whether this effect of IL-6 receptor blockade really has implications for the adipose tissue mass expansion that we see in response to tocilizumab. It is possible, at least. And currently, I could maybe mention that we are about to finish a study where we address the role of IL-6 by using, again, the IL-6 receptor antibody in the postprandial substrate turnover. So in this study, we will focus on more, first of all, on the postprandial substrate turnover, but we will also focus a bit more on tissue-specific effects of IL-6. So we are going to take or are taking tissue biopsies from fat, abdominal fat, and from muscle, skeletal muscle. And we are also assessing the arteriovenous differences across muscle and adipose tissue. So we hope really soon to have a much better and broader overview of the role of IL-6 in regulating substrate metabolism and how it may be regulated or how it may regulate adipose tissue mass. So all the people on this slide have in some way contributed to the data I presented, and I would like to highlight Becky Trin. She's a fantastic MD PhD student, and she performs all our quite invasive stable isotope studies. And then the training intervention study, which I also presented data from, was performed by Louise, Regitze, and Anne-Sophie. And they were all MD PhD students at the time, and they did really an amazing job to carry that study through. Also thanks to funding agencies, and thank you for your attention. And then I'm happy to discuss with you. Thanks very much.
Thank you, Helga. Wonderful, and brings up a lot of mechanistic questions for us here. How does it affect re-esterification, and why is the lipolysis not affected? Are you going to, wondering if you are going to investigate some of this in a cell system? Would you be able to take human cells, grow them in culture, and evaluate some of these mechanistic things in cells, or Is that not a direction you want to go?
Yeah, I mean, I wish we had taken this approach already to begin with. So this was our first study using stable isotopes, and we wanted it as simple as possible. So we did not take tissue biopsies, unfortunately. So now I feel like we should repeat it all and then get those biopsies so we can study, in vitro or ex vivo what really happens within the tissues. But what we can do is to work with human, both adipocytes and also skeletal muscle cells in vitro. And we are currently just establishing those things very slowly. I mean, they exist in our center, but not within the group so far. But we are getting into that, and that is really crucial if we want to understand on a more mechanistic level what takes place. So right. Yes, we will do that.
Great. So a quick question before I get to the Q&A. Does IL-6 affect mitochondria at all? Does it do anything to mitochondria?
One of the things that we really wanted to do in this study that I mentioned, the postprandial study, was to do Ouroboros or respiratory measurements from both muscle and adipose tissue. Following with and without tocilizumab, with the assumption that yes, possibly that IL-6 would affect mitochondrial respiration. But to my knowledge, I mean, no one has studied it in this, I mean, in such a context where you use tocilizumab or in a human setting using like an IL-6 receptor antibody. But I wouldn't be surprised if there would be, some effect, but like with IL-6, it can always, it's a very pleiotropic molecule, and it can really act in so many different ways, and very often it surprises you with opposite of what you hypothesized. Yes,
that's true. So Anna is asking, very interesting talk. Do you have any thoughts on how this affects COVID patients, as the drug apparently is used in their treatment? Is that true? That this might reduce systemic inflammation, but would it possibly cause increased visceral fat?
Yes, that's a good question. And yes, it is true that that COVID-19 patients are treated with tocilizumab, and they present when they are critically, I mean, when they are ill enough, I would say, and hospitalized. IL-6 levels are sky high in these patients. I mean, I talked about, 5-fold increases in exercise, and then you reach 5 picograms per mil. But in these patients, you are way above 1,000. So it's a different scenario. And yes, treating them with tocilizumab will reduce inflammation. CRP levels drop probably back to baseline. And I mean, whether that will influence visceral adipose tissue mass, I would say yes, on the long term, it probably would. But I imagine that these patients are given a one shot of tocilizumab that lasts for a couple of weeks or a month or so. And under those conditions, we have no evidence to say that will change body composition in any way. So the study that I presented was a 3-month study. So we also don't know if you treat for longer, is that a continuous effect, or is it something that flattens at a certain point?
Right. Okay. Related question by Stephen Cornish. Blocking IL-6 would have a significant effect on the cytokine signaling network. Do you know what signaling mechanisms would be interfered with when blocking IL-6? For example, is TNF-alpha increased more, or any other?
It's a very good question. So we have both, when we have infused IL-6, we have often looked at what does it do to other cytokines. And in those studies, I mean, I would say we did short-term infusions, an hour maximum. And under those conditions, we did not see other cytokines that we measured, the typical TNF, IL-1, IL-1RA, not IL-1RA, IL-1, IL-10, TNF-alpha. They were not changed in response to IL-6 infusion. And then what we observed in the study I just presented was that when we infused tocilizumab, on the acute day, TNF levels rise. So as soon as you block IL-6 very acutely, within half an hour, TNF levels were elevated and remained elevated throughout the study day. And interestingly enough, when the participants came back on the last study day, 20 days after the infusion, the TNF levels were back to normally, they were normalized again. So other than TNF, what did we measure? IL-1, we did not see anything. IL-8, also nothing. TNF was the only one that we observed and responsive.
Okay, good. Helga, I think we're going to have to move on. I very much appreciate your talk, and very informative, and lots of questions come up.